Thanks everyone again for joining this Space Force meeting. On behalf of myself and Jeff Weil and Pallav Kasuri from Salk Institute, we're the organizers of this meeting. And so today we've got Jason Kim from 10x to speak. Jason is the Senior Science and Technology Advisor for San Diego and Orange County of 10x, and he's going to talk to us about 10x Genomics solutions for spatial analysis of gene expression in situ. So over to you, Jason. Thanks.
Thank you for the introduction, Peter. And can you see my screen?
Yeah.
All right, excellent. So thank you, everyone. And apologies, I am, all you're going to see right now is a background, and that's mostly because my young daughter is not feeling well, and as much as she's trying to give me some space to do my talk, she sometimes has to run in here. And so to keep it from being a distraction, I thought it'd be probably best to just keep the camera off for the moment. So again, thank you to the organizers of Space Force for allowing me this wonderful opportunity to talk to you guys today about our Visium spatial analysis solutions. And I usually like to start my talk with just kind of a little bit of a musing about some of the things that I see when I read the many papers that I do. And earlier this year, and this is actually a paper, if you hear me talk quite a bit, I've referenced this as a preprint for quite some time, but I'm really excited that this paper was published in Nature Neuroscience earlier this year. And it's actually a really cool approach to sort of what I think ambitious possibilities for spatial analysis. But this group published this paper, and what they did was they created this topographical gene expression map of the human dorsolateral prefrontal cortex of the brain. What they did was they took serial sections from this area of the brain and performed whole transcriptome mRNA sequencing analysis on each of these sections of tissue. They then used a spatial gene expression profiling approach from 10x Genomics that I'll talk about, but they were able to maintain the physical properties of the tissue while also getting that mRNA-seq analysis done. And so what they did was they took these data from these serial sections and they stacked them together and stitched them, basically creating this 3D spatial gene expression map. So this is the topographical gene expression map that they themselves sort of refer to in this endeavor. And within that data, they were really able to confirm a lot of these canonical gene signatures when they compared it against the Allen Brain Institute's Brain Atlas data. But what was really key was that they discovered a significant number of very new, novel cortical layer enriched genes that really were not previously identified in any of the sort of the big brain atlas databases, including the Allen Brain Institute. And studies such as these, where that physical location information can be retained in traditional mRNA sequencing, is what's really driving the excitement and hype around spatial gene expression analysis. So when you hear spatial, the word spatial, sometimes you could it may be one of those things where it's a word that may not truly convey the power of the technology or the underlying sort of methodology that's going on within that particular approach. And so let's see, click next. But to take a step back a little, this concept of spatial analysis, it's not new, and certainly 10x Genomics didn't invent it, right? It's a technique, a field that's been most notably in decades old to current histology, histopathology, clinical pathology, and techniques like immunohistochemistry, FISH, there's RNA-scope, newer things like small molecule FISH or single molecule FISH, RNA scoping, like with different types of additional multiplexing targets and other chemogenic or fluorescent staining assays. They're still in wide use today and very powerful tools. And certainly if you know what you're looking for specifically and the targets number in the small handfuls, these are going to be very useful and informative assays. There's even abilities to go into cellular and some of these other things that I mentioned, subcellular and even single molecule resolution. but all of them have some limitations, right? There's always that talk about, there's pros and cons, strengths and weaknesses to everything. And again, if you know what you're looking for, and that number of things you're looking for are going to be in general limited in number, at least per processing event, you're looking at low plexity, and something that's targeted, right? Again, you have to know what you're looking for. But again, you're going to have some trade-offs. There's some trade-offs with signal to noise, precision, and the fact that a lot of this information is mostly qualitative in nature. And then on the flip side of things, there's a whole host of molecular techniques. And more specifically here, I'm referring to next generation sequencing. It's probably an understatement to say it's been revolutionary since its general adoption some little over, a decade ago or so. And with NGS, you essentially get everything data-wise. It's just supercharged discovery, whole genome, whole transcriptome, whole exome, total RNA-seq, epigenome, you name it. It's been or being done. But one of the big trade-offs with this, at least in the context of this general discussion, is that you completely lose all spatial information, right? You're taking that DNA or RNA or whatever that source material, and you're extracting it from the tissue and losing all of that, even in single cell work. So the thinking here is, how can we combine the strengths of both of these approaches, something that retains all that spatial information from the tissue, but utilizing all that powerful data encompassing, discovery powerhouse of next generation sequencing. And that's what spatial transcriptomics at its core aims to achieve, and why it's such an exciting and highly popular approach. As I mentioned before, in molecular assays, in most molecular assays, typically you're going to take a piece of tissue or biofluid, whatnot. It'll be dissociated either into single cells or completely ground up to get access to the DNA or RNA contained within. And mostly, at least in terms of the spatial context, it's a completely destructive process, but you do get tons of data. And that data is obviously of extremely high value because of this inherently destructive nature, you lose all locational information on where a particular set of data was ultimately sourced from in physical space. Spatially, you could imagine having two different samples of the same tumor, let's say, with nearly identical single cell types, right? So let's say you dissociate both these tumors from two different patients, and you do a cataloging of the cell types that are there, and the abundance can be found in that you'll see probably the same cell types and cell numbers in general between the two tumors, but without that spatial context, you wouldn't know that maybe one of these samples or these tumors had most of its tumor infiltrating lymphocytes, for example, at the periphery of the tumor, while the other had them located within. And that could have key differences or impacts to how, downstream therapies or other interventions could play out. And so that just highlights how important having that spatial information is. When we talk about 10x Genomics and they say, who or what are you guys? And what's nice is, I was chatting with Peter earlier, and I've been with the company for about 3.5 years, and I've said the explanation of who 10x is in various different ways over the years, and it's evolved quite a bit. And luckily, a lot of people know about us now, and they know of us to be not just a reagents or assays or application company, but we do have a focus with all of our products to provide as seamless of an end-to-end solution as possible. And so, it starts with instrumentation, of course, our assays and different various solutions, and ultimately onto software analysis. And, sort of speaking on instrumentation on the single cell side, which we're not really going to talk about in this particular forum, but we recently announced the launch of the next generation of single cell analysis, the Chromium X and iX system, which will essentially make high throughput millions single cell runs sort of a routine, normal part of the future. So really excited for that. But, if you think about where we've come as a company, we're now over 2,300 publications in just single cell genomics and transcriptomics. And, we are a provider. I think it's it's an understatement maybe to say that we're one of the most trusted, if not the trusted provider for single cell sequencing and analysis. And, you know, let's be real here, that this is probably what we're most well known for, but we're also very much well known for our exciting spatial gene expression solutions, where, again, as I mentioned in kind of that earlier story, you can perform mRNA-seq on that intact section of tissue for both fresh, frozen, and now very recently we've made availability to do this on FFPE tissue. And so that's what I'll be covering in greater detail as part of my talk today. So what I'd like to introduce to you informally is that, or formally I should say, is that this is the Visium spatial gene expression solution. And this is a way for us to be able to take intact sections of tissue and perform a full mRNA-seq on that section of tissue while keeping that spatial information intact. So now you would now know not only what cells or what genes are present, but where exactly they are present. And this opens up new ways into approaching sort of that data analysis. And if you're very familiar with single cell analysis already, you're probably very familiar with this t-SNE or UMAP plot here on the left, which is sort of the bread and butter of single cell analysis. And you can approach very similar as you would that sort of same analysis by using cell or spot cluster information and take a cluster of cells with a particular gene expression pattern of interest and trace those back to where exactly on the tissue they were physically. So here, let's say I'm interested in this cluster that shows increased levels of several genes of interest of mine, and using the spatial data, I know they were all localized in this area of the mouse brain. Sort of on the other side of things, I could take an image first approach, something that's very similar to how a pathologist approaches sort of the tissue image, and analyze the tissue image itself, looking for a particular area or areas of interest. I can then highlight the area and be able to do a full report of genes that have increased or decreased levels of expression in that particular location. I can even compare areas of tissue section to one another, or even across sections of tissue. And now I'm able to perform what is essentially molecular pathology by doing differential gene expression analysis of the full transcriptome from any area or areas of the tissue, and even between sections of tissue comparatively. And because all the underlying mRNA sequencing information is collected from that entire section of tissue or whatever's placed on the Visium slide, I can essentially go back and do multiple analyses post-production on any and all areas of the tissue section without having to go back and sort of recollect data from new areas of interest. And how this technology works is that we take a special DNA barcoded oligos, sort of similar to the 10x gel bead, if you're familiar with that, and spot them onto an area on a glass slide. So you can think of kind of that 10x gel bead sort of smushed onto a glass substrate. And each spot has millions and millions of oligos, each having the same unique 10x barcode. And this barcode will differ from spot to spot on the glass slide, and it is known which spots have which barcodes. So very similar to the single cell technology, but where the 10x barcode in that context was used and considered as the cell ID, here in the Visium assay, the 10x barcode can be thought of as the locational ID. So again, since all the barcodes and their positions on the slides are known, you know that all sequencing reads that share the same 10x barcode will all track back to a specific and unique location on the glass slide. So for instance, if it's, you know, barcode 123, you know that all the reads that excuse me, have that same barcode will all have sourced from this area. And then you can look at the subsequent tissue to see where that fell physically on the tissue itself. On the VisionOnSlide workflow and the way the slides and samples are prepared is that, just like in a histological preparation, a section of tissue is placed on the capture area of the Visium slide. So the Visium slide actually has 4 capture areas. It is then stained with H&E or immunofluorescent probes. The H&E is essentially used as a morphological map, so you can kind of see where what the structure of the cells in the tissue are, and then the underlying gene expression information underneath. Next, a high resolution image is captured using a microscope. Again, that image will be serving as the map, and then this same stained section of tissue will be permeabilized to get access to the mRNA, which is then captured by those 10x barcoded oligos on the glass slide that I just spoke about previously. Barcoded molecules are then generated, the tissue is dissolved away, and those labeled cDNA molecules are then collected and processed sort of through standard Illumina NGS RNA library prep. And so, still images and illustrations can only detail, so much. And so what I'd like to share is a quick 30-second video that animates this technology a bit to provide some more context. And I should mention up front that there isn't any narration going on. So if you're not hearing any music or any sound, it's just there's just light background music, but this is just kind of so that you can see how the technology works in a more animated fashion. All right. Oops. All right, next. There we go. And whether it's fresh frozen samples or FFPE tissues, the Visium workflow is very similar to our single cell assays in that it's completely end-to-end and is complementary to Illumina next generation sequencers. The assay, what we provide is the Visium slides and necessary reagents to spatially barcode the RNA and perform NGS library prep. Once sequenced, we have both a primary analysis pipeline and visualization software available. So there's no real upfront capital equipment costs, at least on the 10x side, to be able to enable Visium. You essentially just need sort of standard access to histological equipment so that you can get your tissue sectioned onto a Visium slide, and then a microscopy to be able to capture those images from the stained sections of tissue. And again, you're going to see a lot of parallels between what, excuse me, spatial and single cell sequencing looks like, but all 10x libraries are immediately compatible for sequencing on any Illumina NGS system. I would say the performance and data quality is pretty much the same no matter what sequencer you choose from. And if you've heard me talk about, which ones we recommend, essentially I give the same advice is that, what sequencer you ultimately choose is one that you have most ready access to at the sequencing price point and turnaround time you're looking for. So, obviously, if you're looking for the best bang for your buck, you're going to want to do a NovaSeq run if you can have. But if you're looking at, let's say, a timetable of months to get your data back versus, something on a HiSeq or, the new NextSeqs, you may need the data quickly, but at a cost. But that's really essentially it's validated on all sequencers from Illumina. So really, the choice is flexible and yours. Additionally, there have been publications demonstrating sequencing 10x libraries on sort of long read sequencing systems, such as the PacBio SQL or the Oxford Nanopore systems. The cDNA that's created off the slides are actually full-length transcripts prior to sort of that typical fragmentation that's needed for compatibility with Illumina short read sequencers. And so it's possible to get full-length transcript information in this sort of modified sequencing approach if needed. I'm not going to go into too much detail about it, but we do have a analysis pipeline and visualization software that's available at no cost. The primary analysis pipeline is actually called Space Ranger, which again is analogous to Cell Ranger on the single cell side. And then on visualization, it still uses that Loop software, which again is also available at no charge. And with this software, you can explore your spatial data and do sort of that low to mid complexity data analysis in a highly visual medium and interface. You can generate those spatially naive plot information that I kind of showed earlier in the talk, do some clustering, do differential gene expression analysis. You can highlight areas of the tissue of interest and get a report out of gene expression in those areas. If you're interested in trying out the software and playing around with the spatial gene expression data, we do have both the software and demo data sets available for download off of the 10x website. They're full versions. We don't charge for our software. And I mentioned H&E being sort of the far more preferred method for morphological staining, but in addition to H&E, Visium is also compatible with immunofluorescence staining. This method allows you for really doing true multi-omic spatial analysis by combining that protein information from the immunofluorescence staining along with that underlying whole transcriptome readout of the tissue section from the Visium probes themselves. And if you are interested in doing IF on Visium, it doesn't really require any specialized or proprietarily labeled antibodies. If you already have fluorescently labeled antibodies that you do immunofluorescence with, for instance, they should be perfectly compatible with Visium. Currently, the 10x software, though, has the capability to multiplex up to 6 antibodies, or more specifically, I should say 5 antibodies plus DAPI. And I should also say the other part of the equation is that your microscope that you have access to should have the capability to see, visualize these different wavelengths. But once you have both of those, you can multiplex a few antibodies as part of the immunofluorescence staining. And as part of that workflow, there is a 10x slide cassette that creates these discrete wells for each of the sample sections on the Visium slide. So that it really allows you to use different antibodies for each sample. So you don't have to necessarily do, one set of antibodies for all the 4 samples that are on the Visium slide. And while spatial analysis by itself is a really powerful tool, many groups have already started leveraging already existing or soon to be generated single cell data sort of in ways that can enhance each other. So I've seen people who are starting with Visium, they're adding single cell data at the same time to help refine spot clustering information that comes from the Visium spatial assay. On the flip side, I've seen groups that have already generated tons of single cell data and are trying to do refining of the spot cluster information with that data, or they've also done attempts at sort of reconstructing or putting 3-dimensional form to single cell data sort of in silico. So they're taking, potentially canonical markers for, certain regions within a particular section of tissue, and then reverse applying that to the single cell data to sort of try to build or build inference maps, 3D maps spatially out of that data. So very, very cool things that are happening on the informatics side that are involving both the single cell and the spatial worlds together. And some examples of this, and I'm not really going to go into these next couple of vignettes in exhaustive detail, but really just more to show the different ways that this trend is really manifesting itself. And I would say that, right now it's almost hard to find papers that are just purely spatial in nature. There's really a lot of folks that are really trying to combine the two, but I mentioned powerful bioinformatic tools that are being built to take that previously spatially naive single cell sequencing data and using spatial transcriptomic information as this sort of gene signature scaffold to try reconstructing that single cell data back into physical 3D form. Sort of like, I think about it like doing paper mache with sequencing reads, right? So you use the spatial information first as the wireframe, and then you're taking your single cell data as these pieces of paper mache that you're trying to reconstruct. And this group actually did create a spatiotemporal analysis kind of in that same vein of the developing chicken heart using both the combined single cell and spatial data. So very, very cool. During the sort of the several time points that they collected, they were able to do a lot of trajectory reconstruction, especially during the development of the and morphogenesis of the heart. And this approach, again, using combined 10x single cell and spatial solutions, but this time on cutaneous squamous cell carcinoma samples, sort of to discover a potential immunosuppression feature. They were able to observe physical co-localization of regulatory T cells and CD8 T cells in compartmentalized tumor stroma. Again, the analogy of sort of, if you think about like a sports team and like basketball players, that what players you have on the on your team, but where exactly on the court are they? Or, when I spoke of the 2 tumor samples that are potentially, if you dissociated them into single cell nature, they could both come out in the same cell types and abundance, but you wouldn't know locationally where they were. And this will allow for, I think, greater fine tuning of being able to find biomarkers for certain diseases and being able to do a lot more precision or targeted approaches and therapies. Speaking of pharmacological therapy, specifically here, looking at acute respiratory distress syndrome, same combined method. The authors here knew that damaged responsive fibroblasts modified the lung microenvironment in some way to promote infiltration by immune cells, but ultimately at the expense of lung function. And unfortunately, this immune response is sort of this double-edged sword in that it clears the infection away, but also at the same time causes self-harm damage to the lung tissue. And there is currently no therapeutic for this. So ultimately, when they did this single cell and spatial combined approach, it helped them identify some potential targets, this extracellular matrix protease activity of these damage responsive lung fibroblasts. And they could be affected in ways to sort of preserve the lung tissue at the same time as improving immune response and ultimately and hopefully clinical outcomes. So again, knowing not only just what cells they're present and in what abundance, but where they are in physical location to one another can now add that extra wrinkle in being able to fine tune this data to be able to target these biomolecules or biomarkers for therapies. And then finally, this group took that combined data approach through a time course of the kidney from early injury to sepsis to recovery. And they created this sort of spatiotemporal profile of each stage along the way. They identified a cell type through single cell sequencing that persisted through the entire timeline, but ultimately, again, they didn't know its physical location. So they were able to identify the cell was there at every point, but just where was it? And so using Visium, they were able to track sort of its location through the various phases. And they found that those cells to be located to primarily the outer stripe of the outer medulla. So it was this blown in area that you see here in this, oops, go back here. Oh, wow, sorry. My mouse is really sensitive. Outer medulla here. And I mentioned earlier that we not too long ago started shipping Visium that's compatible for FFPE tissue. What you just saw in a lot of these publications were with the fresh frozen version, but we're really excited to have finally made FFPE available or compatibility Visium available. So as you can imagine, if you sort of look at our technology, the probes, the oligos that are used for capture on the Visium slide are primarily very same construction as our single cell gene expression assay, where they utilize a poly-DT capture for the polyadenylated mRNA. And so typically what you need is full length or intact, non-fragmented pieces of mRNA molecules to be able to be captured. But obviously in FFPE, because of the fragmentation and cross-linking that's inherent in this process, that was sort of a barrier in being able to utilize that method to be able to get good data from FFPE samples. And so the approach for FFPE is actually very, very interesting and just a slight wrinkle on the fresh frozen version. So it still uses the same Visium slide and spatial barcoding technology. But for FFPE, instead of targeting and capturing the mRNA directly using that poly-DT capture, we use a method called RNA Templated Ligation, or RTL, which involves using a probe pair against the protein coding transcriptome. So one half of the probe pair has a proxy poly A tail, and the other probe pair half has the sequencing primer. Each gene has one probe pair, and the probes then hybridize to the target sequence. And only when both of them are matched do they ligate to one another. And by this method, it's sort of like think about a lock and key or other analogy using a pairing and validation system, is that by this method, we minimize any non-specific ligation and reduce off-target effects. So after RNA templated ligation, the RNA is digested away, releasing the ligated matched probe pair, and the proxy poly A tail is captured by the oligo DT on the Visium slide. If only one of the two probes is bound, capture by the 10x oligos would not occur, right? Because you don't have this, this probe that has the synthetic poly A tail, or sequencing would not occur because you don't have the sequencing primer that's on the other pair. And so either way, it would not proceed to data generation. And so you can, again, the analogy I like the best for this is I think of it most like two-factor authentication from a website. For like your banking website, you need to have both the password and the secondary factor authentication to be able to get in. And we currently have transcriptome probe pair sets for human and mouse containing 18,000 genes and 20,000 genes respectively. So this is our RTL probe approach for FFPE. And when you compare the two workflows overall, they're really very similar. The primary differences lie in the staining and capture section of the process. So in fresh frozen, the cells of the tissue are simply permeabilized. So if you look at the top here, they're just simply permeabilized. You saw in the video and in the sort of the first diagrams of the sample prep. And the mRNAs then within those cells are allowed to be captured by probes on the Visium slide. They're barcoded, the tissue's digested away, and then you move forward. With FFPE, there is that additional step because of the RTL probes that I just detailed, that then the cells become permeabilized, and then the RTL probes are captured by the oligos on the Visium slide. So you can see here sort of an additional sub-step that happens right here with the probe hybridization and ligation. When you look at the overall workflow, it looks like this for FFPE. And if you're already sort of familiar with the fresh frozen version, you'll notice immediately here that there are a lot more stopping points built in than before. This should really give much more flexibility in processing options and logistics, especially if you're leveraging sort of a histology core or maybe a histology lab for doing those upfront steps. And as you can imagine with FFPE, of course, there's going to be a deparaffинization and decross-linking step that's not necessary with fresh frozen tissue. So overall, though, the two workflows in general follow the same sort of general theme, but kind of to boil it down to the technical comparisons of the two versions, highlighting sort of, again, these major differences, at least from a technical standpoint. You can look at it kind of like this. The fresh frozen here on the left, Visium for FFPE on the right. The capture method is the primary difference. So in fresh frozen, we're directly capturing RNA through the poly A tail, through the poly DT probes, whereas on Visium for FFPE, we're capturing the products of the probe pair, the RTL probe pairs. Off-slide biochemistry, where we're making the libraries, we're looking at, because you're creating that full-length cDNA, you do need to prepare it for short read sequencing. So there's the fragmentation, you ligate your adapters, you do end repair, ligate your adapters, and then you do your PCR for your sample indexing. Whereas in the FFPE version, it's actually a little bit more streamlined because you are just taking this RTL product that, again, one side has the sequencing primer already on it. So you're basically just going to need to do single index or the sample indexing PCR, and then you'll be able to go straight away into sequencing. The big takeaway here, I would also say, is because on the fresh frozen side, again, you're dealing with the actual transcript information. You're going to need more sequencing because you are going to have to align those sequences to a reference transcriptome, and then you have to have a confidence interval to make sure that you're truly seeing coverage in the transcriptome in certain areas to do your accurate transcriptome counting. So it does require a little bit more sequencing. I would say, if you think about it, just the back of the envelope, thinking your sequencing burden is probably half for FFPE than it is for fresh frozen. So because you're sequencing just that shorter probe pair bit that's with the FFPE RTL probes, you don't need to sequence nearly as much as you do for the fresh frozen. And thinking about sequencing, how much do you have to sequence? I know that that's a very common question for single cell, is that, again, this is primarily for fresh frozen with the 50,000 read pairs, but the idea here is the same. You're wanting to sequence based on the percentage of tissue that's covering the Visium sample section. So this doesn't have to be super specific. You don't have to go in with a program to try to get it down to the point percentage. Just kind of eyeball it and maybe overestimate a little. But here's some examples. You see this is maybe 75% of the area, 50%, 25, etc. And what you're going to do is you want to take, there's 5,000 total spots in this capture area, and you're going to want to multiply that by the percent tissue coverage, and then multiply that by 50,000 or 24,000, depending on whether you're doing fresh frozen or FFPE. And that'll give you the sort of targeted number of reads for the sequencing. So in this example, if there's 50% of tissue captured, and let's assume that this is fresh frozen, so you take 50% of the tissue coverage times 50,000 or 5,000 total spots for times 50,000 read pairs per spot for 125 million total read pairs. So if you were doing 100% of the, let's say you have a tissue that covers 100% of the spots on the tissue section, you're looking at 250 million total read pairs maximum. On the FFPE, what I didn't mention on the fresh frozen side is that typically to dial in the permeabilization and whether or not the tissue will actually work with Visium, we strongly recommend a tissue optimization step, which essentially just dials in the time that you would permeabilize your tissue to be able to get the mRNA captured for your tissue type as possible. We don't actually require that on the FFPE side. There is no tissue optimization. However, there are some things you want to do up front to qualify whether or not your tissue will be successful and work with Visium. And the number one thing we recommend is doing a DV200 analysis. So we don't recommend RIN because obviously this is degraded and fragmented RNA. It's not sensitive enough of a measure for FFPE RNA quality. What DV200 does is, if you're not familiar with it, it's just a percentage of RNA fragments that are greater than 200 nucleotides. And this is a much better predictor of FFPE RNA success with NGS assays than RIN. And so the idea here is that you want greater than 50%. And you can have high confidence that that sample should work with Visium for FFPE. Now, that isn't to say that below 50%, you're just going to see a complete failure. It's just that you will start to see potential impacts to data. There's no guarantee that there's going to be an impact to data. It's just that you potentially could see some let's say less fidelity in your data quality by going below that number. And so it's one of those things that maybe you might want to do sort of a small pilot study if your samples are of lower DV200 percentage quality, or maybe even just try to select different blocks that are 50% or greater. The other thing to consider also is if you've ever worked on the histological side with FFPE sections, you know that tissue adhesion is a big challenge. And so we do have a tissue adhesion QC test that essentially is just, you'll just section sections of tissue on FFPE onto this slide, and then go through sort of that first few steps of the process where you're going to be essentially doing the water bath, deparaffinization, all this other steps, and then testing to see if the tissue just adheres to the slides or floats off. In histological approaches, right, there's a lot of ways to sort of combat this. There's charge slides, superfrost slides, but the Visium slides are neither. And so you just want to make sure to just check that your tissues are not just going to completely float off of the Visium slide when you start processing. But let's say you've gone through, you've done your DV200 and you've done the tissue adhesion test. You did both of those. You can go in with a high degree of confidence that your tissue should be compatible with the Visium assay. We also have done a lot of testing in-house of various different types of tissues, both on fresh frozen as well as FFPE. This is right here, just an example, not an exhaustive list of ones we've tested. On our website, though, you can get the current most up-to-date list of tissues we've tested. We've tested human, mouse, rat, both on normal and different various common disease tissue types, just to show differences in sort of what can and can't work. There are additional things. I would say for the most part, we've had a good long while Visium has been out to see what tissue types have worked successfully. I'll say that if you're working on pancreas, if you're working on salivary glands, anything that has strong enzymatic activity, you'll probably want to take due diligence and talk to your science and technology advisor or FAS from 10x on sort of different approaches, because we've had conversations with others who have done it either successfully or at least it's a elimination activity of eliminating what absolutely does not work. The other thing to take additional consideration or discussion with us is if you're doing anything bone related, because there's obviously a decalcification step and potentially can be impacting downstream data quality. So if you're working with either of those tissue types, I would say, maybe reach out to us and let us have a conversation in more detail. But for the most part, I've seen this work well on pretty much every other tissue type out there. So we do have a lot of resources for Visium available on the 10x website. We have recommendations on, the sectioning. So if you're doing fresh frozen, you're going to need a cryosection, cryotome. If you're doing just regular FFPE sectioning, just a regular old microtome, it will be fine. You will need some way to visualize and take images of the stained tissue. So access to microscopy and image capturing capabilities. We have all kinds of protocols, white papers, demonstrative protocols, videos, etc. for both. And so just a wealth of information there if you want to do self-service. You also, there's human support, both remote and on-site. So we have a fantastic technical support team, but also us. So I am your local 10x science and technology advisor. Actually, I think I see a lot of familiar names. So if you've come to 10x talk, you probably heard me speak before. As you know, I'm always available to chat about your experiment and what you want to do with our technology. But we also have great field application scientists who are very, very well versed in analysis, special sample preparation, and things of that nature. So with a group, the take home here is that you have a team that is fully dedicated to support you in every way possible. And then sort of the one last thing I want to mention is that we've hosted, as part of the launch of the Visium FFPE, a series of webinars that were focused specifically around that product. So there was a series of 3. So you'll think about it as, the first one was sort of a high level information on the product itself, one's focusing on sample prep, and then one finally focusing on data analysis. And so these are all past. So I would say they are available on demand. So it's actually kind of nice in that now you don't have to mark a day on your calendar. You can actually just, whenever you want to take a look at any of these, you can go to this link and view the sort of the talks completely on demand. I will say that I highly recommend attending these, even if you've kind of got the gist of the technology through my talk, primarily because the speakers in each of the sections are involve a person from the product development team, and often they're able to provide some unique insights to the product and its capabilities that, obviously they're going to have firsthand experience and knowledge with. So I think there's some interesting insights that they can offer that probably is unique to them. And so finally, just as a kind of a wrap up, our Visium assay for fresh frozen sort of comes in 2 options. Again, I mentioned it briefly, but we have a Visium tissue optimization slide and reagent kit. This is no NGS. It's a more economical way to be able to dial in that optimization time for permeabilization. It actually just uses a fluorescent readout, but once you've dialed in your time for permeabilization, you're then able to go on to your Visium spatial gene expression slide and reagent kit, which actually does create the libraries that you will use for spatial gene expression sequencing. And then on the Visium assay for FFPE, again, it does not need tissue optimization, but you will need to select based on species, whether you're working on human or mouse, because remember, there's RTL probes that are involved for that. And if you're interested in more details on either of these components, available pack sizes, options, pricing, quoting, please reach out to your sales exec, and you actually have a new one that I'll provide contact information for in the next 2 slides. But just to kind of wrap up, again, thank you so much for this opportunity to speak to you today. I've worked with Brian James, and I've worked with a lot of you folks throughout the years, and so it's always an extreme pleasure for me to be able to come and speak to you about any of the new technologies of 10x Genomics. But again, this is a really popular and exciting assay that's gaining a lot of buzz and momentum, primarily because it doesn't require any capital instrumentation. You can just, if you already have histological capabilities, you can begin generating that high resolution spatial data immediately. Its primary advantage, of course, as sort of same with any mRNA based whole transcriptome based assay, is that it's a discovery tool, but it's especially powerful as a validation tool as well, especially when you're coupling it with protein information. And because you get data from every part of the tissue that's mounted on the capture area, you can really go into your work with that confidence that you're not really leaving any of that data behind. And so I want to, again, just kind of close out with our contact information. You can take a screenshot. I'd be happy to send this your way. But if you're looking for pricing, please reach out to Omar or Sonal. My contact information or Susie's is here. If you have any other additional questions about the product or any of the sort of capabilities or things that I've talked about today, I'd be more than happy to share any additional information as I have. So with that, again, thank you so much, and I'll kind of hand it back to you, Peter.
Great. Thanks very much, Jason. That was really helpful and informative. Appreciate it.
Yeah, thank you.
And I think we've got some questions coming through. I know Jeff Weil has a few questions. So go ahead, Jeff. Others, just put questions in the chat, and I can read them out.
Thanks very much, Peter. Thanks, Jason. And I'm going to have to go right after you answer the questions, because I just found out about a problem at home I have to attend to. So one is, I was hoping you would briefly address this issue of sensitivity, because it seems to me that with 5,000 spots, again, you are not going to be able to achieve single cell resolution. If I'm wrong, correct me. If I'm not wrong, tell me what the true resolution really is. Secondly, because of the mechanism by which your strategy works, and I do appreciate the fact that we don't have to put out money for any capital equipment, but the way that this technique works is that there is a diffusion step that's required to get material out of the cell and onto the slide, which raises the question of how much lateral diffusion and contamination in adjacent sections there is. And I'm curious about if anybody has done an assessment of that. Other techniques such as seqFISH Plus or MERFISH are direct detection in single cells. So could you please address those 2 points?
Absolutely. And I'll try to keep it as brief as possible so that you may be able to take off. The spots, you're correct. They are not single cell-ish. So they're 55 microns in diameter. So we say it can be anywhere from 1 to 10 cells, depending on the size of your cell. So if you're working on let's say large neuronal cells or other large cells, it could potentially be 1 or 2 cells per spot. If you're working with small immune cells, it could be, 10, 15 cells per spot. So you're correct, which is the reason why a lot of people are merging single cell data with Visium data to try to infer or deconvolute more of that spot information. But to be completely transparent, it is an informatic approach, and there are potential limitations to that. To address sort of the your second part of the question is that lateral diffusion, actually, I was surprised to learn when we purchased Spatial Transcriptomics as the company that had this, they actually had about 10 papers that were already published in Nature and other publications that did test the lateral diffusion. That was like the #1 thing that I think most people were concerned with. And if you permeabilize correctly, this is the reason why we have that tissue permeabilization step, which is you want to dial in that you're not over permeabilizing, that you now start getting lateral diffusion. You obliterate your cells to the point where everything's flowing all over the place. But you want to open them up enough that you get as much capture of the mRNA as possible. What the studies have shown is that if they dialed in that permeabilization time correctly, that the lateral diffusion is actually 1 to 3 microns, which I'm going to be honest, like it surprised me that it was not as much as I had thought. And so there's a dead space that is inherent in the design, not because of the diffusion, but just because the probes can only be stuck close as possible. And so there's a center to center distance of 100 microns. So there's a little bit of a dead space. And if there is any lateral diffusion, again, with the asterisk of that optimization for permeabilization has been done, the diffusion should not cross over to where you start seeing bleed out of data into neighboring spots such that it convolutes the data even more. And I can send that paper to everyone in the group just to have that as a sort of a read and reference.
Okay, thanks very much. Appreciate it.
You're welcome.
Olivier has a, I'll read it out. Olivier, I don't think can unmute. So it's Olivier Harris-Mendy. Is FFPE commercially available? Which core facilities have Visium instrument on the Mesa?
Yeah, so FFPE is commercially available. It started shipping, I think now we're a month past, so it's already been in the hands and it is shipping. So if you order, you can get your hands on it. I can send a list of which cores and facilities have capabilities right now to do Visium that's relatively in the area. I can also break it down to sort of a full service versus a semi-full service. So there are full service where I give you tissue and my Visium slides, and you do the cutting, mounting, processing, give me data. There's also some that are, you slice it and put it onto the slides and you send me the slides and then I can process them. So I will send both options to the group.
But presumably when you say which cores have facilities, I mean, what we're talking about is a fairly conventional histopathology facility and a sequencing facility.
That's correct. That's correct. And when you boil it down, some are offering them as kind of a concierge to have the logistics taken care of.
Okay.
But yes, you have a histology access or microscopy access. You really don't need a facility to necessarily do it.
So I guess in a way, what Olivier is asking is, because in principle, there's probably loads of places on the Mesa that can do this, but maybe what Olivier is asking is in practice, which kind of pairs of histopath facilities and sequencing facilities are actually doing this routinely?
Okay.
Do you have that information? People on the Mesa.
Yeah, I will send that over.
Okay.
Yeah.
If you send that to me, I can forward it to Olivier and anyone else who's.
Sounds good.
I don't know if not sure if you know Olivier.
I believe I do.
Yeah, you've probably met him. Yeah. I had a question. Couple of questions. So presumably with this slide format, I mean, we could put multiple pieces of tissue on one area of a slide to get, basically to decrease the cost per sample. Is that right?
That is correct. That is correct. So there is actually a couple papers that did that, where they took, I think, mouse spinal cord sections, and I think they did 4 or 5 on a section. So it's almost like a very small tissue microarray for more of a brute force sense. And so, yeah, absolutely. There's no additional need, because the spots almost act as a sample barcode, because locationally where they are. So there's not really much by way of deconvolution that's needed in the data either. Right.
And you mentioned combining it with immunofluorescence.
Mm-hmm.
So what I wasn't clear from that was, can we, based on the immunofluorescence data, then can we define regions of interest based upon single or multicolor immunofluorescence, and then analyze the expression profiles within those regions of interest selectively?
That's correct.
Okay, so that kind of analysis is compatible with Cell Ranger and Loop, is it?
That's correct. Yep.
Okay.
Yeah, you can either do it in an automated fashion, or there's also, a manual hand drawing, sort of like how a pathologist would annotate with markers on a slide. But you can do it any of those sorts of ways. Yeah, definitely.
Okay.
I have a question.
Sure.
Go ahead.
Yeah, I'm wondering. So, I know Fei Chen published this paper relatively recently on using a computational method to sort of deconvolve the different cell types that might be combining on each bead in your setup. Have you used that a lot? Is that compatible with your platform? And if so, how well does it work?
Yeah, so I would say there's a lot of different computational third-party tools that are available to do that type of work. I think much with every informatic approach, there's going to be pros and cons to them, right? Because of the way that inherently they build their algorithms to make inferences. Some will just do straight out canonical genes and then try to assign those to a sort of a qualitative score, a semi-quantitative score to suss out the number of cells or the cell types that are there. It's compatible with our data in that if you have single cell data from the same tissue type and spatial data, any one of those informatic tools can be used, and that's kind of what I was showing with some of the papers there, is that some of those are using their own in-house tools. Seurat, that is a very, very highly used single cell third-party tool that's been published on just a mind-boggling number of papers, has that capability and feature as well. So yes, I guess the short answer is it is compatible with our system and our data. Our software, though, directly does not do that inherently by design.
Got it. Thank you.
Excellent.
I have one more question. So this is maybe more relevant to the fresh frozen than the FFPE, but I'm wondering about microbiome infiltration into tissues. Can you detect that by sequencing?
That would be difficult. So now I'm going to talk about probably something that's more that I enjoy, which is out of the box sort of ideas. So with the advent of the FFPE solution, it's shown that you can kind of, almost very similar to single cell, design your own primers going in to maybe potentially target certain things that are not necessarily intended to be targeted. So you potentially could. The challenge, I would say, is you might just lose most of the at that point, you'd be probably kind of doing something similar to semi-bulk, right? Because, you would know that let's say you are able to detect them. And now you're able to detect them in which spot. But beyond that, you wouldn't be able to get too much more information outside of qualitative metrics, if that makes sense, because now you're going to kind of see, okay, I see a lot more sequences for, you know, this particular bacteria in one spot, you know, 10-fold times than somewhere else. So you just know in general there may be more there. You just wouldn't have the level of resolution that that I think that most people would want in a, let's say, a more single cell type of resolution, or at least in a compartment that allows for maybe more quantitative analysis. So yeah, I think your two barriers there would be trying to design a RTL-like approach to the bacteria that you are focusing on.
Well, I guess that's why I was thinking, wouldn't it be more compatible with the cryo sections where you don't need the RTL?
Well, the problem is that unless it's that just only captures indiscriminately poly DT, anything that has a poly A. So yeah, so
right, fair enough. Okay. Well, there's no more questions in the chat, so unless anyone wants to put something in quickly, and in that case, yeah, if you could send us information on, you know, facilities which are running this routinely, Jason, locally, that would be really helpful.
Absolutely. And I just shared the screen back again with my contact information. You know, if you think about anything later on, or you are wanting to discuss anything, I know that sometimes right at the cusp of, especially at the end of the day, you know, the questions might come, you know, later on. Don't hesitate to reach out to me. I'm more than happy to chat and talk answer any of the questions at a later time as well.
Yeah, will do, Jason. Yeah, thank you very much. It's been really helpful.
Thank you. Appreciate it. It's always a pleasure, and I hope to see you guys in person at some point in the near future.
Yeah, thanks.
Thank you.
Bye.